Hello everybody, welcome to this webinar which is on food allergen testing. Has it got more complicated? This is one of a series of webinars that we have been putting on, but the Anaphylaxis Campaign have been putting on for everybody during these very interesting times. This webinar has been significantly oversubscribed, which shows the popularity of the topic and indeed of the speaker. And I would like to introduce to you Richard Fielder. Now, Richard is a very old friend of the Anaphylaxis Campaign, and I think I've known him since I joined the campaign 12 years ago. He's a very active member of our corporate food panel, and he's always there to give advice to people because we get a number of queries to our helpline around these issues, and Richard is always there to help us with those queries, which is great. I'll just tell you a little bit about Richard before the webinar starts. For the past 30 years, Richard has worked internationally for leading diagnostic test kit producers, providing advice on immunological and molecular techniques. With this experience and being a qualified microbiologist and a BRC third-party auditor, he's been an active member of the food allergen community. And in fact, his biography says he's been actively engaged since 1996. He looks too young for that. Richard's been an active supporter, as I say, of the Anaphylaxis Campaign for many years. So without further ado, I'd like to hand over to Richard so that he can give his presentation on this really important topic. Thank you, Richard.
Thank you, Lynn, for those kind words. I can assure you I've got the scars and wounds to prove the long service in the industry. Hopefully I can share that with the audience today. So thank you indeed to everyone who's registered for the webinar, whether you choose to watch it now or to watch it later. I'm pleased to present on the topic of food allergen testing. Normally we don't get quite as many people as this tuning in for such a topic. Maybe it's the question I'm posing, and perhaps it won't be a spoiler to perhaps answer that question right at the beginning and say, yes, I do think it has got more complicated, and hopefully I won't lose too many people now by explaining in the next 30 minutes or so how and why I believe that is the case. The style of my presentation will be a series of quite detailed slides. I'm not expecting that I will have the time to go through all of those slides, all the details on the slides. I'm putting them more for review for later to provide the evidence. What I will try and do is provide a commentary to answer this question and give some personal views on the different aspects and the overall question. So without further ado, we shall move to the first slide. So let's just, oh, there we go. Okay, so in true Ask a Millionaire style, I'd like to start by asking a few questions of the audience, just to help me understand the type of organisations which might be present today and how much time they spend on this topic. So if we can launch the poll, please, Megan. There we go, and on your screen now you should see these questions in a poll format, so please go ahead and select the responses you feel are the most appropriate. Sorry, it's quite difficult to phrase a question to capture all type of organisations, so but it'll give us a feel of the interest of this particular group today, and indeed how much time they spend on this, let's face it, quite a specialist topic in people's probably normal working day. There we go. So let's have a quick look now. There we go. So we're saying 48% of people on the webinar today are working on gluten-free products. The next biggest category is vegan foods. So that is that's sort of following a certain trend, which we're going to report on. And indeed, we've got a lot of labs and research organisation present. So I do appreciate my colleagues in the Anatol community joining me, and hopefully I represent their views as well as my own today. And I think if we look, actually surprisingly, a lot of people here do spend an awful lot of time considering food allergen analysis. So, okay, that's quite different to a conference we held in back in February. So that's helpful to know. So I'll just close that. And let's move down to the first slide to outline. So today, my presentation will be in 4 parts. The first part is really trying to address the context. So what's changed for allergen testing? Then I shall move on to what you need to know before you start testing. That's not necessarily as a new user, so it should be applicable to all. And then we'll move on to looking at how to ensure reliable results, but and lastly, look at the possible impacts of new technology. In all of this, we're coming back to the question we started with, which is, has it got more complicated? So that's the context. I'm trying to frame these 4 elements today. I think we are all used to, in our working lives, looking at the context of the organisation and assessing the risks, the opportunities and the threats. As food manufacturers, when we follow the GFSI standards, there's a management commitment to do so. In ISO under 17025 for laboratories, we need to look at the context, not least for business contingency planning. And certainly with ISO 9001, 2015, again, sections 4.1, 4.2. So I think we're all familiar with doing analysis. Hopefully you're going to reach the same conclusions I have. So just to explain how I approach this subject in my working life, we look at the opportunities, risks and threats. We do a sort of mind mapping process. We look at that in terms of different scenarios and how different trends are developing over the course of time, and look at the consequences for the different stakeholders we work with. And that's obviously to lead to an action plan. But this is a continuous process. The world is continuing to revolve around us, and we have to update and review this constantly, often by the second. Things change that quickly. And I think that's really the context we've got used to in the past with these business models. It's been a fast moving and complex environment to understand. But today, I think perhaps these plans don't fully convey the times we're in. And the third dimension I would add is that disruptive element. We've seen it with the pandemic, but I think there's other bigger issues also which are evolving, climate change being just one. And then we have other issues which are perhaps more minor irritations, panic buying. You know, these are all influences which are disruptive by nature. The reason why I explain the methodology we go through is just to relate you back to food allergen analysis. The fact that whatever approach we take, it might well be in some way not flawed, but it might have some limitations. So there are some systemic, if I can get the words out, errors. There are also some random errors. And ultimately what we find depends on who does the sampling and analysis, what is sampled and analysed, how the sampling analysis is conducted, and how often it is performed. And these are all common themes which I'm going to echo today in terms of food allergen analysis. And just to say that testing is only ever one piece of the jigsaw in food safety and quality. And it's really, really important to view the whole picture, because otherwise we can get false impressions. And if time had been greater today, I would have actually run a little advert from 1986, which the Guardian newspaper created. It's quite iconic, but this is basically the text from that, which if you get time, have a look at that, because I think it puts things back into perspective about how we approach and analyse even the broader landscape. So in terms of, has it got more complicated? Well, yes, I think it has. I think where we started, and that's always a point, if we look at the starting point before the pandemic, then we saw that fundamentally a number of things were happening, and there was a growth in plant-based diets and free-from foods and vegan foods. That was already taking place before the pandemic. And also within within analysis, there was a paradigm shift in the types of technology we were using for testing. Broadly in three areas: on-site testing, mass spectrometry, and with molecular techniques, next generation sequencing. So it was already going to be difficult and complicated introducing new technologies with a broader range of food products to a wider audience, because there are increasing number of people which need to follow an elimination or an exclusion diet because of food allergies and sensitivities. But in more recent times, following the pandemic, we've now got the disruptive influence of all the COVID measures. We have, as a result, a global recession as people and countries have had to go into lockdown. And that's at a time when we're trying to launch new products, seek new ingredients. We seek meat-based plant-based meat substitutes, plant-based dairy substitutes. So this whole area of plant-based diets is coming at a time when it's very difficult, when supply chains are disruptive, when our normal processes of evaluating and securing new supplies and assessing new raw materials would take place in an environment of close cooperation. And that is obviously hampered with social distancing and other measures. In addition, the global recession makes the potential for food fraud more on our radar in terms of a risk and a threat we should be looking at. We know that product recalls from food allergens is one of the, if not the leading cause of global food recalls. And we've seen a number of scientific developments in recent months and certainly years as well in terms of practices for food allergen management, thresholds coming in. We've seen changes in the accreditation in labs to improve and to focus resources on method, validation and verification. So in terms of analysis, the scope of methods, sampling, method validation, method verification, all these factors are the topics which we need to discuss today in order to ensure that we can supply safe food of the stated quality, that the claims on the packs can be trusted by consumers, and that the precautionary allergen labelling is minimised. So what's important to know before you start testing, whether that be from the perspective of doing it for the first time, or probably in most people's situations, they are launching new products, and perhaps they're also reviewing their testing regimes to increase capacity to cover off and mitigate new threats. So whatever the reason is, we shall have a look at some of those basic things we need to do. I have asked people over the course of this last year what areas of food allergen analysis they find difficult. So wherever you see this survey finding symbol, hopefully I remember to introduce some of those feedback. So 5 things to know before you start. Sounds like a parent here, do your homework. Certainly is something we have to say to our boys. But what I want to do is just mention briefly 5 elements which I think is really worth getting to grips with before you undertake your testing. And that starts with the risk assessment, looking at the resources we need. Food safety culture, I could have mentioned earlier and perhaps should have done, but we know it's a new element in the new GFSI standards such as BRC. And when that has an impact on all sectors, all areas of an organisation, and particularly testing as well. So we'll come to that. Obviously, we're going to look at appropriate methods and ways of sampling, and we're looking at the claims and stuff which we might finally have on a product. So let's look at these 5 elements. And we start with the risk assessment. This really has to be the starting point for testing because it is defining all the priorities, which then maps out the risks and what we need to do about it, the order in which we do it, and the amount of resource we need to devote. The higher risks we should devote more priority to. And indeed, in the survey findings, we do find that people are considering testing at this early stage. And also that what they are doing is devoting most of those resources to the higher risks. I'm just a little bit conscious of time already, so I will try and keep this fairly brief. In terms of supporting resources, once we've won the management commitment and the financial commitment, we need to look at the resources we need. Not every situation does lend itself to installing testing. Some environments and workflows within maybe factory areas for having a small testing area do make it very difficult, particularly when, let's say, there's aerial contamination. What is important is that people who do testing are trained and they need to really understand the science and the purpose of what they're doing. And ultimately, they need to understand that actually to do sampling and testing well, it is expensive. In terms of the culture, it is important because we're wanting buy-in, and we're not only in the technical departments, it's all departments, including purchasing, to make sure that actually the right methods and the right test kits are used in the right cases. So one size fits all or one supplier fits all might not be appropriate. Also, we can't categorically guarantee the absence of an allergen. We talk about detected, not detected. Therefore, we need a culture of critical thinking. We must always be trying to question assumptions and the detail behind numbers, how they've been built up. We can't ignore opposing viewpoints, what we call selective perception. We must always be questioning in order to avoid false impressions. Sometimes it's convenient, perhaps, that the evidence all fits. Certainly in a lot of thrillers it is. But really, we've got to try and look beyond that because often sampling can mask the truth. Just if we got very quickly, selective perception, just to give you an instance of what I mean. If I show you this picture and tell you it's a picture of a bird with its beak to the right, some of you may believe me. Whereas if I actually tell you it's a picture of a black cat with its back to us, looking over its left shoulder, that is the actual situation. So that is selective perception. Next point I want to get onto is sampling, because there have been enormous changes in detection technology in the last few years, which in the next slide we will go through those. But other areas of analysis, particularly sampling, the analytical community are particularly concerned that our understanding of the theory of sampling and its application is actually, there's a big gap here and there's more work to be done. And in the analytical community, there are efforts and initiatives being launched, such as this one. And indeed that correspond, well, actually, with the survey, what we found is actually a lot of people believe they have documented procedures, trained and qualified people to do sampling, and they have a level of confidence in their approach. So therefore, I think there is some work to do in this area to actually communicate more about our knowledge. Moving on then to the quite impressive choice of techniques we have these days. As Lynn said, I started back in 1996 with one test kit. Now there are test kits for most allergens and not all, and some are for on-site testing and others are lab-based. The ELISA technique is the gold standard method, but what we do find is that results will differ between the different test kits, and therefore in proficiency rounds we report results per test kit or per test kit method, and even we will see variation within test kits on the same sample. One of the biggest challenges I see is with these paradigm shifts in technology is the introduction of on-site tests, because often they are, well, without doubt, they are very simple to perform, but they're actually one of the most difficult types of tests to introduce and implement effectively, basically because the environment in which we are trying to use them, as opposed to a lab environment where we've got a quality management system, which is all tuned around method validation, the scope of methods, methods verification, where we've got trained analysts who understand metrology, the science of measurement, and a number of issues to really help interpretation of results. So, and this is indeed what people do find, is that on-site testing to implement it is difficult. Coming back to this really crucial point, this is why testing has got more complicated, because we're trying to provide more foods and claims on packs. We need to be sure that they are correct. And at the end of the day, analysis can only determine the presence. It cannot guarantee with categoric assurance the 100% absence. So terms such as free from are for consumers clearly understandable marketing terms, but for analysis we talk in terms of detected and not detected presence rather than presence or absence. And when we talk about presence, it comes with constraints, with limitations, with defining exactly what we mean. And we normally use limits of detection for on-site tests, and for the lab tests, they are quantitative, so limits of quantitation. And because these claims push us to trying to assure some level of guarantee that an allergen is absent, then we are always working at the limits of performance of a method, down at which is always going to push the method to extreme. And I would always recommend that it's worth looking at the method scope to make sure you're selecting the right method, and then look at the validation reports and certificates of analysis. Looking at the numbers for limits of detection, LODs and LOQs on a piece of paper doesn't give an idea of a method performance. Right, we've been going 25 minutes as I look at my watch. So in the next 10 minutes, I will try and go through actually what's quite a difficult section, but so I'm probably going to have to limit this because of my ambitious nature to include as much as possible. But here, testing has got more complicated because we need to provide these label claims. We need to provide precautionary allergen labelling only when it is needed following a thorough risk assessment. In order to do that, we need to recognise that sampling is really important in the whole analysis from sampling to decision, and actually most of the variation is takes place in these initial steps. If we were talking about mycotoxin testing, it would be 80 to 90% of the analytical variation is with sampling and sample preparation. Reliable results, at the end of the day, this is a question of uncertainty, or in other words, the variation around results. And in order to improve accuracy and precision, we must decrease uncertainties. And these uncertainties which come from systemic errors in our approach, in our methodology or in the lab, which lead to bias, or they also come from random errors. So while we might hit the target, our precision is very poor. So it is inherent in all measurement to have a measurement of uncertainty. And what we must do is try and take every step all along the way to reduce that in order to produce a more reliable result. So that leads neatly on to 10 places where I believe there is the potential for analytical issues. And if we look at the 10 parts of the analysis I've listed here, the first 7 are to do with sampling and sample preparation. And perhaps that does help to reflect where the efforts needs to be put first and foremost to get to reliable results for supporting safe food of the stated quality. So as time is against us now, I will have to rush through some of these slides, but do look at the details. Scope of the method, I think it's vitally important that first of all, we do check that the method we're going to use has been validated for the very types of samples. That then means all we have to do, I say all we have to do, is to verify that we can achieve the same performance with our samples, and we can demonstrate that we are competent at performing that analysis on a repeatable basis. If it's not within the scope of the method, then we have to conduct more extensive work. And this is whether it's validation or verification. When we take our risk assessment and we looked at characterizing our hazard, when we look at processing, if we're looking at a finished food and whether it's been processed, that is going to have a marked influence on our ability to detect the target allergen. Next, I've already indicated that sampling is extremely important, and I think these 2 guides, one by Camden, Guide 78, makes the case very clearly, which sample is meant to represent the population, and inevitably it will be an imperfect representation. I think once we have taken that as a starting point, what we see, and these are the words from the Royal Society of Chemistry, is to look for appropriate sampling. And what they mean is that we're linking sampling with the question we're trying to answer. Is it fit for purpose? So therefore, we can have different styles of sampling programs to suit different purposes. But within all of this, we do need the person who's doing the sampling to be competent. And that's really important when we've given this ability in the form of on-site test kits to users. So we have sampling plans and we have sample types. And this gives a construction of sampling plans and the different types of sampling. A lot of people we're finding will perform selective and random sampling plans coming out of their risk assessment. The general advice is trying to avoid composite samples as we do in microbiology. Composite samples are often used, but microbes will multiply. Allergens don't multiply on their own. In terms of sample types, the performance of any method is determined by the sample we're trying to analyse, and there are such an infinite variety of types of samples to analyse. And we shouldn't forget that retained samples are important. They always give us the ability to go back, and that is very useful. When we look at sample types, what we're looking at is the diversity, we've mentioned that, whether they're being processed, because that affects our ability to detect them. And we look at their composition. We look for cross-reactivities, and we remember from 2015 the recall with cumin and the cross-reactivity of Marhaleb in almond-based ELISA kits. And we also look for matrix effects, and that could be particularly important in this new context where we have new plant-based foods and we've got new formulations and new ingredients, and seeing the effects on analysis. So that's really important. Swabbing, I think we could probably have a whole webinar on this. It's such an important topic because one of the principal controls is prevent cross-contamination is cleaning intervention. And what we know with swabbing, it's often a very variable technique with very low recoveries. And we're talking about, with these two references on here, anything between 0, I do mean 0, and let's say 30%, in that sort of region. And we also need to consider where we are doing our swabbing, or we're collecting even rinse samples. With allergens, we are not concerned about disinfection, the removal of bacteria. We're interested in the removal of allergens. So we should be doing our sampling after the intermediate rinse, after the surface is cleaned by the detergent. And there's lots of good reasons to do that. Something for another time, I think. Samples, if we look at this sample here, we can see there's bits and pieces in it. What we're trying to achieve is homogeneous samples, and that's really difficult. If we do achieve homogeny, we often end up with a paste, and whatever equipment we use, that then gives us the double difficulty of cleaning it afterwards and the potential for cross-contamination. So sample preparation contributes to analytical errors. It's difficult to achieve homogeneity, and it's also difficult to clean the equipment afterwards. If we look at the test portion, some work done by PepsiCo a few years back now, but this is really opens people's eyes to the test portion, the actual amount used in the test. And this was looking at gluten analysis, I think in oats. And what they're saying is it's not a normal distribution when you do repeated analysis on a batch. And actually what you get is a skewed assessment. So what they found was when a gluten kernel exists and provides an overall average of 20 milligrams of gluten per kilo, roughly 2 out of 3 observations will end up less than 20. That's something to think about. So smaller samples, more difficult to achieve homogeneity. So that's going to increase analytical variance. So something to think about. We know that people find extraction very difficult, and the more complex the food, the more difficult it is to separate out the other components which could affect our analysis. Usually these are fats. And this is just showing some examples of, we can see separation in our extracts. It doesn't mean to say that we can go on and reliably detect with these tests, but it is a first stage to actually being able to detect, is getting the allergens into solution. So what number is this? This is number 8. So we've done all the sampling and sample prep questions to where errors might creep in. When we're looking at ensuring reliable results, we must also look at using reference materials. Unfortunately, there are very few, but Monica have several milk reference materials we can use, and LGC this year in February just launched a reference material containing milk, egg, almond, hazelnut and walnut in a kit. So those are materials definitely worth getting your hands on and using in your analysis. And so our proficiency samples, these are samples which are homogeneous, which have been analysed by laboratories and have a consensus value for that material. In the case of gluten, this just illustrates with a homogeneous sample analysed by the same method by 90 labs, that's the range of analytical results which was observed. What's more important to you is your samples and trying to simulate the processing conditions which they might have undergone. So preparing your own incurred materials, not easy, but is always worth doing. And it's something as a test kit manufacturer, we're doing a lot of other companies are also doing the same. So it's very important, as well as using spiked samples as well. They all tell you something. And something to think about also would be doing a material balance like we do in the factory, a mass balance, to ensure reliable results, which is just making sure that the inputs and the outputs match. So the kits and the analysis done do align themselves. Reporting results. We are trying to move towards, well, first of all, we're trying to move away from parts per million recording because for traceability reasons, the units of mass is the kilogram. And in terms of allergens, then in the clinical sense, it's the protein which is responsible. So we are trying to report results as a community as milligrams of allergen protein per kilo of food, and where we don't do that, to provide conversion factors. Sounds simple, but that's also a topic for another day. Interpreting data, this is often quite hard, and often it is about trying to understand the question. And just to give you an example, I haven't used this example for a while, so I thought I'd throw it in again. If a bat and a ball cost £1.10 together, and the bat costs £1 more than the ball, how much does the ball cost? My wife got this right first time. I didn't. I shall tell you at the end. So I'll give you a few minutes to think about that one. Lastly, I think this really is so important to the industry, trying to minimise precautionary allergen labelling. And it does mean interpreting clinical reference data, so the dose, which is responsible for eliciting a reaction, and understanding what level of protection we can provide. Vital is one scheme coming out of Allergen Bureau in Australia and New Zealand, and has been updated recently. One of my colleagues, Rachel Scott from another company, Roma Labs, they did a webinar recently with Ben Rebington going through this, which I should urge you also to look at, as well as this excellent paper which Ilse just brought out, which had a very high participation of UK talent, I would say, from FSA and also from RSSL. It's not easy data to translate threshold data into action limits. We are looking at taking a dose and converting it to a concentration and then relating that to a serving size. Basically, what they normally find is people often eat twice as much as the designated serving size. So that does also need to figure in our risk calculations. But if we look at the different methods, it is very difficult to analyze with these higher portion size, 500 grams being a ready meal. So those were 10 things to think about. Basically, you've got to be looking at every step of your analysis, trying to minimize that analytical variation to get to a reliable result. And we've got some challenges. Technology drives change. It's meant to make our lives easier. Often it makes it a lot more complicated, and we have to think about a bit more about how to use it. And that's definitely the case here. Great potential. I mean, I entered this business because I love the change in the technology, but we are all going to have to devote more resources to sampling and analysis, not least because we are going to be working with new materials. I think the most commonly used proteins in plant-based diets are soya, grain, peas, nuts, legumes, mushrooms, and even insects. So I know insects don't really figure in our method validation and verification. So, and also we need to look at the new analytical technology to see how best to use it. It's great, really great that we have such a wealth of tools in our analytical toolbox, and they each need to find their own role. Particular interest to me and perhaps concern is how we can support the implementation of on-site tests. I think a lot more is needed in this regard. And I think also we need to change our perception of how testing is used, from telling us what has happened to why it's happened, what will happen, or what's the best or worst that could happen. So I think testing and the use of larger data sets is helping us, will help us to move beyond where we are now, which is telling you what has happened. So in conclusion then, I do think that it has got more complicated. I think that the market and the need for such plant-based foods, for free from, for vegan, is really important, and that there are a number of challenges needed to introduce testing technology and have a robust testing regime. And I would remind everyone, do your homework. Remember, sampling determines what you find or don't find. There's no one size fits all here, unfortunately, and always assess test performance with your own sample types. At the end of the day, consider the bigger picture. So if you'd like to take part in the survey, I put the link on there, and I hand back to Lynn. Sorry, we only have 3 minutes for questions. So let me.
Thank you very much, Richard, for that excellent presentation, which had so much information in it. And I'm sure some of those slides merit a webinar in their own right, as you said, but it gave us a fantastic overview of the issue. So thank you so much for that.
Perhaps I could just pick up there, actually, because I had meant to say, Lynn, actually, they do merit another webinar. Indeed, you know, it might be the chance just to say with vegan foods, I think you have plans for another webinar on that, don't you?
Yes, we do. And we're finding these webinars are so popular. The level of engagement is very high. So clearly there's a demand for them. But I think we might be coming back to Richard with a webinar on some of the specific issues raised. We've had lots and lots of questions coming in, and we're not going to have time to do them all, inevitably. However, what we will do is put a Q&A on our website. So if you have sent a question in either in advance of the webinar or as we've been having them during the webinar, we will make sure we have those answers there on the website, and we'll pull those together with Richard. Also, the presentation, I mean, what's been very impressive for me watching the stats is that everyone who joined the webinar at the beginning has stayed with the webinar. So you clearly are saying some pretty important things, Richard, too.
That's a first.
You know, nobody, the webinar, again, will be put on our website later so that people can actually watch it again or pass it on to colleagues. So that's fantastic. I'd like to get a couple of questions in, if I may, Richard. And as I say, we'll get in as many as we can, but don't worry, the other questions will appear on the website. So one of the questions we've been asked is as follows. With ever increasing sensitivity of tests, lower and lower amounts of the allergen are detectable. DNA testing gives a positive negative result, but no quantification, but is biased towards particular species. So true risk is difficult to assess, for example, cod in fish. How close are we in the UK to setting threshold levels? And if we do set them, how will we assess risk if we are testing for a species that's not 100% recovery in ELISA? There's a question.
There's a question. Lots of.
That sort of issue is reflected in many of the questions we've received.
Yeah, I think the topic of, I'm not surprised allergen thresholds has come up. I think in the UK we have led the debate on allergen management and use of thresholds. I think the FSA would give you the more definitive answer on this. However, what I can say, the reference I put up, the ILC funded study, Chun Hun, I know from the FSA, was involved as one of the authors on that paper. Also that there is a European project which is being led by Claire Mills in the UK, Professor Claire Mills at the University of Manchester, the Thrall project. And it is, to answer that question really, trying to look at not only the quality of the clinical data, but also to look at reference methodology so we can assess risk. And part of what we're involved with in the analytical community through a group called MFAM, which is linked in with that project, is to look at ELISA and mass spectrometry methods with 6 different allergens: egg, milk, soya, almond, hazelnut and walnut. I think the 6 are. So correct me, anyone, if I was wrong with those 6. So I think there is a lot of work being undertaken, and I think this is where schemes such as Vital will be important. I think the Europeans will look for their own system. At the moment, we have different allergen thresholds in 3 member states: Germany, the Netherlands, and Belgium. So there is a need to move towards consensus, and I would suspect that North America will find another solution. Though at the end of the day, we all look at the same clinical data, and experts work internationally. So hopefully we can get some harmonised approach to this.
Thank you, Richard. And interestingly, thresholds is an issue that's often raised by consumers to us via our helpline as well. So clearly is a hot topic. So thank you for that very clear answer. I'm going to squeeze another one in.
Okay.
Historically, carryover risk in cooking oil has been considered low due to the protein being denatured. There have been recent suggestions that the denatured proteins may still be a risk, but they're not detectable using standard tests. Is there any up-to-date information on this?
Well, yeah, processed foods, I mean, generally are always difficult to detect proteins because they get denatured or in some way affected by the processing. I think oils are a special category, really. I mean, I remember probably 15 years ago now, I remember some work from FARP and the late Sue Heffley. She used to consider aspects of carryover in cooking oils and whether the methods we have will detect the proteins in the oils, whether because a lot of the extraction methods we use, say for ELISA, are aqueous-based methods, and making sure that actually the proteins which are present, whether they are soluble in those aqueous phases, and we're not excluding those which are perhaps hydrophobic and in the oil layer. So I think it is problematic. Funnily enough, last week we did tweet some work done by the gluten watchdog about carryover of gluten to fries from breaded products, and indeed they were finding there was indeed carryover and reporting levels. This was taking from I think about 60 different, yeah, certainly a number of takeaway fast food outlets in California and Ohio, I think it was. So that study's worth looking at. And I think there have been other methods developed by the FDA looking at sort of protein-based detection. But I think this is where mass spec would be extremely helpful in understanding it. But there are practices which people are using to try and you know, segregate the cooking oils for breaded and non-breaded products, for example. Not reusing oils, filtering them, all these things do help to minimise carryover.
Thank you, Richard. I've got time to get some more in. We're making you earn your keep today, aren't we? The next question. What should we be doing to reduce the possibility of cross-contamination? Gosh, that's a broad one.
Oh, lots. I think, yeah, it probably comes back to your risk assessment. I think, you know, if you make sure that it is well designed or well considered, the risk assessment, and obviously considering all the different risks, and then to try and document it, and then and review, I think that will help it certainly will help. And I think the role of testing is really to layer in that evidence to support whether controls are effective or not. I think broadly testing should give the objective evidence, but it's only one part of it. So auditing, you know, and monitoring and all the other good aspects of control, scheduling, segregation, you know, these are all things which would help.
Thank you, Richard. Another one that I've got several here. How frequently should we be swabbing equipment to test for allergen residues? Should it be done between every production run or sporadically?
Yeah, I suppose this comes back to making sure that the design of the clean, you know, will fulfill what it should do, which is remove certain targeted allergens which have been highlighted in the risk assessment. So I think, yeah, design of is first of all using testing to make sure the design is right. And then, and at the same time, making sure your verification test is used at the same time as you do validate. Make sure you repeat the clean several times to make sure you've got, that you've established you've got a robust cleaning regime, which does remove the allergens of risk. And I think what that study should also help you do with the risk assessment is tell you where you know you will have problems. So what we're trying to do is challenge the clean to see whether it is effective. So we want to look at the difficult to clean areas, not the 10 by 10s on a flat surface. We want to look at a filler head or a mincing plate. We want to look in tanks behind spray balls or the bottom of vessels where the scavenge pumps might not be working. These are the areas where, you know, if you have access, you would want to try and swab. But I think also we have to highlight, you would want to cover perhaps over a course of time some of the other areas. It's just that part of the risk assessment and the cleaning validation will establish where exactly and the frequency with which you need to test. And actually, you know, the scheduling of a clean can also determine how much, you know, whether it's a deep clean or an interim clean. All these are. So I think Peter Littleton, who we know very well from Chris Daines, and he's a member of the corporate board. He will I think he's going to do a webinar on this topic. So I think, you know, these are the people, you know, as test kit companies, we can help, and as laboratories, we can help, but also you need in here a mixed team. And I think this is where the hygiene chemical companies could really help as well. So Peter.
Yeah, as you say, we've got Peter lined up for a webinar in the not too distant future. So again, we'll be able to go into that in a lot more detail, but thank you for that overview, Richard. I'm going to give you one more, and this is quite a broad one, but I think it really is an important one, and it's, what are the differences between in-house testing versus laboratory testing, and is one superior to the other?
I try and duck away from saying one technology is better than another or one approach is better than another. I think the truth is we need both. And I think when we do a risk assessment, sorry, I have to come back to that because it really is, you know, it's the sort of the backbone to where we should test and how and why and what methods we should use. I mean, it really depends on the purpose. I mean, you know, for screening on-site tests, yeah, they could do a great job. But if we are validating, then you know, as I say, a cleaning validation, then we should use an accredited lab and we should use laboratory methods. If we were doing an investigation, you know, such as with the cumin recall, then we need to use mass spectrometry, we need to use the molecular techniques as well, you know, or if we're, you know, if the purpose is for official control, then public analysts, you know, would be needed, people with special skills. So I think, yeah, there's no, you need different people for different jobs. And I think that's the thing I love about working in this industry, because there's so many people with different skills and you can learn all the time. So, which makes it hugely enjoyable.
That's a really nice note to end on, Richard. Thank you so much for giving your time this afternoon, for the brilliant presentation, for responding to the questions. This has been recorded and it will be available on our website. There will be a Q&A on our website as well, so people will be able to dip back in, because it clearly is a really complex topic. But thank you so much for helping us this afternoon, and thank you to everybody who is listening as well. And I hope you gained a lot from the presentation, which certainly I think was fantastic. So thank you very much, everybody.
Yeah, thank you for listening from me as well. Thank you very much.